Hello friends, today we are going to discuss overview of prokaryotic and eukaryotic cells. In today's lecture, we shall discuss about the cell in general and structure and function of prokaryotic and eukaryotic cells. Now at the outset, we will talk briefly about the cell. Cell is the basic unit of organization or structures of all living matter. Cell is defined as a unit of biological activity delimited by a semi-permeable membrane and capable of self-reproduction in a medium free of other living systems. Cells can be classified into two families. Prokaryotic cells do not have a distinct nucleus, while as eukaryotic cells have a distinct nucleus. Eukaryotic cells are structurally and functionally more advanced than prokaryotic cells. The terms prokaryotic and eukaryotic were suggested by Hans Rees in the 1960s. The prokaryotes consist of the bacteria and blue-green algae. The eukaryotic cells are found in all multicellular animals, plants and fungi. They have arisen probably a billion years after the prokaryotes and are much larger and complex. Organisms can be classified into prokaryotes, which are made up of prokaryotic cell. Eukaryotes are organisms made up of single or multiple eukaryotic cells. Eukaryotes can be further classified into two families: protozoa, single cell eukaryotes; metazoa, multiple cell eukaryotes. The body of all living organisms, bacteria, blue-green algae, plants and animals, except viruses, have cellular organization and may contain one or many cells. The organisms with only one cell in their body are called unicellular organism. Example: bacteria, blue-green algae, some algae, protozoa, etc. The organisms having many cells in their body are called multicellular organisms, plants and animals. Now the brief description about prokaryotic cell. A prokaryote is a single-celled organism that lacks a membrane-bound nucleus, mitochondria or any other membrane-bound organelle. The word prokaryote comes from the Greek pro, before, and karyon, nut or kernel. Prokaryotes can be divided into two domains, archaea and bacteria. In the prokaryotes, all the intracellular water-soluble components, proteins, DNA and metabolites are located together in the cytoplasm, enclosed by the cell membrane rather than in separate cellular compartments. Classification. Carl Woese in 1977 proposed dividing prokaryotes into the bacteria and archaea, originally eubacteria and archaebacteria, on the basis of structure and genetics. Now the size, shape and arrangement of bacterial cells. Bacteria come in a puzzling and exciting variety of size and shapes. Depending on their shape, bacteria are classified into several varieties: cocci, bacilli, vibrios, spirilla, spirochetes, actinomycetes and mycoplasmas. Bacteria sometimes show characteristic cellular arrangement or grouping. According to the plane of cellular division, they are called as Diplococcus, Streptococci, Sarcina, Staphylococcus, Tetrad. Bacterial structure. The outer layer or cell envelope consists of two components, a rigid cell wall and beneath it a cytoplasmic or plasma membrane. The cell envelope encloses the protoplasm comprising the cytoplasm inclusions such as ribosomes and mesosomes, granules, vacuoles and the nuclear body. Cell wall. Beneath the external structures is the cell wall. It is very rigid and gives shape to the cell. Chemically, the cell wall is composed of peptidoglycan, mucopeptide, peptidoglycan and murein formed by the normal acetyl glucosamine and normal acetyl muramic acid, alternating in chains cross-linked by the peptide chains. Embedded in it are polyalcohol called teichoic acids. Outer membrane. Outer membrane is found only in the gram-negative bacteria. Its function as an initial barrier to the environment and is composed of lipopolysaccharide and phospholipids. The lipopolysaccharide present on the cell walls of gram-negative bacteria account for their endotoxic activity and antigen specificity. Cell membrane. It is a barrier that separates the cell from its surrounding environment. This outer boundary of the cell is also called the plasma membrane. It is composed of 4 different types of molecules: phospholipids, cholesterol, proteins, carbohydrates. Now, the fluid mosaic model of membrane structure. Biological membranes consist of proteins inserted into a lipid bilayer. Integral membrane proteins are embedded in the membrane, usually via alpha helical regions of 20 to 25 hydrophobic amino acids. The hydrophobic side chains of these amino acids interact with the fatty acid chains of membrane lipids, and the formation of alpha helix neutralizes the polar character of the peptide bonds. Proteins can also be anchored in the membranes by lipids that are covalently attached to the polypeptide chain. Cytoplasmic matrix. Cytoplasm is everything inside the cell, excluding nucleus. Cytoplasmic matrix or cytosol or intracellular fluid is the liquid found inside cells. It is separated into compartments by membranes. The main function of cytoplasm is that it is a site of many vital biochemical reactions crucial for maintaining life. The cell expansion and growth takes place in it, provides a medium in which the organelles can remain suspended. Cell reproduction, protein synthesis, anaerobic glycolysis, cytokinesis or some other vital functions that are carried out in cytoplasm. Inclusion bodies. Inclusion bodies are nuclear or cytoplasmic aggregates of stainable substances, usually proteins. Nucleoid. The nucleus is not distinct and has no nuclear membrane or nucleolus, and the genetic material consists of DNA. The nucleoid, meaning nucleus-like, is an irregularly shaped region within the cell of a prokaryote that contains all or most of the genetic material called gonophore. The genome of prokaryotic organisms generally is a circular, double-stranded piece of DNA of which multiple copies may exist at any time. The length of a genome widely varies, but generally is at least a few million base pairs. Now, the prokaryotic ribosomes. There are about 10,000 ribosomes which make up to 30% of the weight of the cell. The bacterial ribosomes are present free in the cytoplasm. The bacterial ribosome sediments as 70S particle, which is composed of 30S and a large subunit is of 50S. They are the site of protein synthesis. Capsule. Capsule is the outermost layer of the bacteria, extracellular. It is a condensed, well-defined layer closely surrounding the cell. They are usually polysaccharide, and if polysaccharide envelops the whole bacterium, it is capsule. And their production depends on growth conditions. Flagella and motility. A flagellum, plural flagella, is a lash-like appendage that protrudes from the cell body of certain prokaryotic and eukaryotic cells. The primary role of the flagellum is locomotion, but it also functions as a sensory organelle, being sensitive to the chemicals and temperature outside the cell. Pili or fimbriae. Hair-like proteinaceous structures that extend from the cell membrane to the external environment are pili, which are otherwise known as fimbriae. They are thinner, shorter and more numerous than flagella, and they do not function in motility. The fimbriae are antigenic and mediate their adhesion, which inhibits phagocytosis. The sexpili help in conjugation. Bacterial endospore. Some bacteria have the ability to form highly resistant resting stage called spores, which helps them to overcome adverse environmental conditions that are unfavorable for vegetative growth of cell produced by certain bacteria from the Firmicute phylum. Now, we will talk about the reproduction in prokaryotic cells. Bacteria and archaea reproduce through asexual reproduction, usually by binary fission. Sociality in prokaryotes. While prokaryotes are considered strictly unicellular, most can form stable aggregate communities. When such communities are encased in a stabilizing polymer matrix, slime, they may be called biofilms. Now we will talk about the prokaryotes and their environment. Prokaryotes have diversified greatly throughout their long existence. Prokaryotes live in nearly all environments on Earth. Some archaea and bacteria thrive in harsh conditions such as high temperatures, thermophiles, or high salinity halophiles. After discussing prokaryotic cell structure and function, let's talk about eukaryotic cells. The eukaryotic cell is much more complex as it contains many membrane-bound organelles to perform specific functions. It contains a nucleus isolated from cytosol and enclosed in a well-defined double membrane. The eukaryotic cells are classified into protists or unicellular eukaryotes like amoeba and algae, fungi which includes yeast, mold, mushroom, plant cells and animal cells. Organization of eukaryotic cell. Let's talk first about animal cell. An animal cell is a form of eukaryotic cell that makes up many tissues in animals and are distinct from other eukaryotes, most notably plant cell, as they lack cell walls and chloroplasts. They also have smaller vacuoles. Due to the lack of cell wall, animal cells can adopt a variety of shapes. Now, the plant cells. Plant cells are quite different from the cells of the other eukaryotic organisms. Their distinctive features are a large central vacuole enclosed by a membrane, the tonoplast, which maintains the cell's turgor and controls movement of molecules between the cytosol and sap. Primary cell wall containing cellulose, hemicellulose and pectin. The plasmodesmata, linking pores in the cell wall that allow each plant cell communicate with other adjacent cells. Plastids, especially chloroplast, that contain chlorophyll, the pigment that gives plants their green color and allows them to perform photosynthesis. Bryophytes and seedless vascular plants lack flagella and centrioles except in the sperm cells. After plant cells, we will talk about the fungal cell. The main characteristic features of fungal cell are as hyphal wall, septum, mitochondrion, vacuole, ergosterol crystal, ribosome, nucleus, endoplasmic reticulum, lipid body, plasma membrane, citizen corpor, Golgi apparatus. Fungal cells are most similar to animal cells with the following exceptions. A cell wall contains chitin, less distance between cells. The hyphae of higher fungi have porous partitions called septa, which allow the passage of cytoplasm, organelles and sometimes nuclei. Eukaryotes are a very diverse group and their cell structures are equally diverse. Many have cell walls, many don't. Many have chloroplast derived from primary, secondary or even tertiary endosymbiosis and many don't. Now dear friends, we will briefly highlight the features of major organelles of eukaryotic cells. First, the cytosol. Cytosol is a liquid part filled inside the cell and it contains water, salt, macromolecules, proteins, lipid, RNA. It has an array of microtubule fiber running throughout the cytosol to give vesicular structure to its destination. Second, nucleus. Nucleus is the central processing unit of cell and homologous to the processor. In a typical computer, the liquid fluid inside the nucleus is called as nucleoplasm. It is a viscous liquid containing nucleotides and enzymes to perform replication, transcription, DNA damage, repair, etc. It contains genetic material, DNA, in a complex fashion involving several proteins, histones, to pack into nuclear bodies or chromosomes. The chromatin in eukaryotic nucleus is divided into euchromatin or heterochromatin. Nuclei are present in a double layer of membrane known as nuclear envelope. Outer membrane of nuclear envelope is continuous with the rough endoplasmic reticulum and has a ribosome attached to it. The space between these two membranes is called as perinuclear space. Nuclear envelope often has nuclear pore, and as per calculation, an average nucleus has 3000 to 4000 pores per nuclear envelope. Number third, the mitochondria. It is popularly known as powerhouse of the cell and is actively involved in the generation of ATP to run the cellular activities. Mitochondria is a double-layered membrane-bound organelle with different structural properties. Outer membrane is smooth and covers the complete organelle with large number of integral proteins known as porins. Porin allows free movement of molecules less than 5000 Dalton within and outside mitochondria. Inner membrane is folded into membrane projections to form cristae. Due to presence of inner and outer membrane, mitochondria can be divided into two compartments. First, in between the inner and outer membrane, known as intermembrane space, and second, inside the inner membrane, known as matrix. Mitochondrial DNA is circular and it has a full machinery to synthesize its own RNA, messenger RNA, ribosomal RNA, and transfer RNA, and proteins. Now the functions of mitochondria. Production of ATP, it helps in the generation of reactive oxygen species in immune cells to kill infectious agents. Chloroplast. Chloroplast is found in plant, algae and other lower invertebrates such as Euglena. Contrasting to mitochondria, chloroplast has outer membrane, an inner membrane and then light pigment containing innermost thylakoid membrane. Outer membrane is porous to the small molecules, but protein or large molecules are transported by translocon on the outer chloroplast membrane complex. Movement of material passed through outer membrane gets into the inner membrane through translocon on the inner chloroplast membrane complex. The inner membrane of the chloroplast further folds to a flattened membrane system known as thylakoids. The photosynthesis machinery such as light absorbing pigments, electron carriers and the ATP synthesizing machinery is present on the inner membrane as integral protein complex. Thylakoid membranes are arranged like stack of coin to form granum. Thylakoid membrane contains photosynthetic green colored pigment chlorophyll. Photosynthesis is an assimilation reaction involving carbon dioxide in water to produce sugar in the presence of solar energy, photons, that catalyzes fusion reaction. Now we will talk about organelles of vesicular trafficking system. The main function of these organelles is to manage the distribution of material, food particles or proteins throughout the cell. Three different organelles such as endoplasmic reticulum, Golgi apparatus and lysosome coordinately work together to maintain vesicular transport of material across the cell. Eukaryotic cell takes up the solid material from outside through a process called endocytosis, whereas uptake of liquid is through a process called as pinocytosis. Similarly, material is secreted out of the cells through exocytosis. In addition, intravesicular system delivers protein synthesized in endoplasmic reticulum to different organelles. Now the first, endoplasmic reticulum. The vesicular network starts from nuclear membrane and separates throughout the cytosol constitutes endoplasmic reticulum. There are two different types of endoplasmic reticuli present in the cell: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum has ribosome attached to it to give a rough appearance, whereas smooth endoplasmic reticulum is devoid of ribosomes. Protein synthesis on ribosome attached to rough endoplasmic reticulum are sorted into three different categories, such as integral membrane proteins, proteins for secretion, and protein destined for different organelles. Now, the Golgi bodies. Golgi bodies are made up of flattened disc-like cisternae arranged in a stacked manner to give three distinct zones. Cis phase receives material or vesicles from endoplasmic reticulum. Medial Golgi is the actual place where protein are covalently modified with a sugar. Trans Golgi is the face of Golgi towards plasma membrane, and this site sorts vesicle for their destined organelles or plasma membrane. Now, we will talk about the third part of this vesicular trafficking system, that's lysosomes. Lysosomes are membrane-bound organelles and an important component of intracellular vesicular system. They are popularly known as suicidal bags due to their role in autophagy. A cellular process probably operates in the cells during starvation to meet their energy requirements. Lysosome lumen is extremely acidic and contains protease, cytolytic enzymes to degrade the ingested material. Now, the centrioles. All animal cells have two small organelles known as centrioles. The centrioles help the cell to divide. Centrioles help in the process of mitosis and meiosis. The centrioles together are typically located near the nucleus in the centrosome. Centrosome is granular mass that is the organizing center for the microtubules. The position of the centrosome within the centriole is at right angles to each other. Centrioles are made up of 9 bundles of microtubules that are arranged in a ring. The centrioles play a major role in cell division. In animal cells, the centrioles play a major role in cell division, but the plant cells have the ability to reproduce even without the centrioles. Now we will talk about the ribosomes. Ribosomes are small particles present in large numbers in all the living cells. They are sites of protein synthesis. The ribosome word is derived ribo from ribonucleic acid and somes from the Greek word soma, which means body. The ribosomes link amino acids together in the order that is specified by the messenger RNA molecules. The ribosomes are made up of two subunits, a small and a large subunit. The small subunit reads the messenger RNA, while the large subunit joins the amino acids to form a chain of polypeptides. Ribosomal subunits are made of one or more ribosomal RNA molecules and various proteins. Ribosome is made up of two subunits. The subunits of ribosomes are named according to their ability of sedimentation on a special gel with a Swedberg unit. Prokaryotes have 70S ribosomes, each subunit consisting of small subunit of 30S and large subunit of 50S. Eukaryotes have 80S ribosomes, each consisting of small 40S and large 60S subunit. With this, we conclude today's lecture on overview of prokaryotic and eukaryotic cells. Hope you have enjoyed the lecture. Thank you.